A study of more than 10 million siblings links birth order to risks from multiple diseases. This is Planetterrian Daily, episode 140. Analysis of sibling data from over 10 million individuals revealed that later birth order correlates with altered risks for several conditions. A large-scale comparison controlled for shared family environments and genetics by examining full sibling sets. Researchers examined patterns across a wide range of diagnoses rather than single outcomes. The work adds population-level evidence that early life position within a family can shape later health trajectories. Follow-up studies may test mechanisms such as prenatal environment or parental age effects. Now, shifting to a very different area of research. A second foundation AI model demonstrated that detailed sleep metrics carry strong signals for future disease incidence and overall survival. The model was trained on large-scale physiological recordings rather than summary sleep scores. Performance held across multiple cohorts and endpoints. The finding reinforces sleep as a high-dimensional data source for risk stratification. Next steps include prospective validation in clinical settings. These high-resolution inputs could refine existing aging clocks that rely on physiological data. On a different note, a new framework describes how fitness, speed, and spatial position interact when populations grow outward, drawing on bacterial colony observations. The models quantify which parameters determine dominance of one group over another. Position at the expanding edge emerged as a decisive factor alongside intrinsic growth rates. The approach bridges theory with measurable spatial dynamics. Researchers plan to apply the same logic to additional experimental systems. From population patterns shaped by early environment to another high-resolution physiological signal that forecasts future disease. Stem cells in mouse models of sickle cell disease displayed functional decline resembling accelerated aging. A drug combination restored stem cell performance and reversed several age-related deficits. The intervention targeted pathways previously linked to cellular senescence. Results indicate that the blood disorder itself drives stem cell deterioration beyond the primary hemoglobin defect. Human translation studies will examine whether similar restoration occurs in patients. This adds direct evidence that disease states can accelerate stem cell aging beyond normal trajectories, from sleep as a predictive data layer to a different physiological system where disease itself accelerates cellular aging. Researchers compared transcriptional control across evolutionary branches spanning 2 billion years. Activation mechanisms remained nearly unchanged, whereas repression pathways showed marked divergence between lineages. The pattern suggests that turning genes on is under stronger conservation than turning them off. Experiments mapped the molecular players responsible for each direction. Future work will test whether the variable repression layer explains differences in gene regulation across species. The work clarifies which gene circuits are likely to be conserved versus rewired during evolution. From disease-driven stem cell aging to evolutionary constraints that shape how genes are controlled across deep time, most people assume that the molecular rules for switching genes on and off are broadly similar across life. The data show the opposite pattern. Activation signals have stayed almost unchanged for roughly 2 billion years. While the signals that silence genes have changed repeatedly as lineages split. Right now, inside your own cells, the same ancient activation machinery is still at work, yet the repression components differ from those in yeast, plants, or bacteria. One memorable number is the 2 billion year span over which this stability and divergence have been measured. The practical takeaway is that efforts to reprogram gene expression in medicine or agriculture will need to account for which layer is being targeted, because the evolutionary constraints are not the same, from conserved molecular logic to another form of neuroplasticity shaped by experience rather than genetics alone. Cryo-EM structures showed that positively charged substrates engage the CLPX entry channel less productively than negatively charged ones. Unfolding efficiency dropped despite intact recognition and ATPase activity. Negatively charged substrates form stabilizing interactions that support successive unfolding attempts. The work identifies charge complementarity as a previously underappreciated regulator of ROP protease function. Follow-on experiments will explore whether cells tune substrate charge to modulate degradation rates, from neural adaptation to a simpler environmental intervention that reduces one driver of insect decline. Studies in mice demonstrate that exposure to infants alters neural responses even in animals that have never given birth. Adaptations appear in regions tied to motivation and sensory processing. Some of these changes are transmitted to offspring through non-genetic routes. The findings expand the view of parental brain plasticity beyond pregnancy itself. Ongoing work maps the molecular mediators that persist across generations. The results highlight how caregiving itself drives lasting neural adaptations independent of reproductive status. Evolutionary biologists tracked fertilization outcomes in systems where sperm from multiple males interact. Cooperative behaviors improved overall fertilization rates compared with purely competitive scenarios. The pattern challenges the default assumption of a universal race among sperm. Measurements quantified how group-level traits influence individual success. Researchers will test whether similar dynamics operate in additional taxa. The work reframes fertilization as a process that can favor collective traits in certain evolutionary contexts. New York University researchers identified the proteins that signal when nitrogen uptake has reached adequate levels. Disrupting these components altered nitrogen use efficiency in experimental plants. The pathway offers a route to engineer crops that require less fertilizer. Greenhouse trials will measure yield under reduced nitrogen input. The work addresses both environmental runoff and farm economics. The identified components provide concrete targets for improving nutrient sensing in crop species. Field tests showed that switching to longer wavelength lighting lowered insect captures at streetlights. The change targets wavelengths that many nocturnal insects detect most strongly. Researchers quantified reductions across multiple orders of insects. The intervention requires only bulb replacement and preserves human visibility. Wider municipal adoption could slow population declines linked to artificial light. The simple modification offers a scalable way to reduce one driver of insect loss. MIT chemists developed a super-resolution approach that localizes molecules to angstrom precision without multiple lasers or specialized dyes. The technique simplifies hardware while extending resolution 3 orders of magnitude beyond standard limits. Validation used both fixed and live samples. The method opens structural studies previously limited by optical constraints. Broader application will test performance across different fluorophore classes. The advance removes a key technical barrier in high-precision molecular imaging. Environmental sampling mapped the distribution of resistance determinants across rivers, treatment plants, and distribution systems. Mobile genetic elements carrying resistance were detected at multiple points in the network. The study links human, agricultural, and environmental reservoirs. Results highlight water infrastructure as an under-monitored conduit for resistance dissemination. Mitigation strategies will target identified hotspots. The data provide a spatial map for intervention in one regional water system. Before we go, keep an eye on further tests of the single laser imaging method across additional fluorophore classes. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. Quick tip from the network. Try Unintended Consequences next. True stories of good intentions that backfired. Every NARA Network show is free at naranetwork.com. And on the website, every episode's visuals live in our free image gallery at naranetwork.com/gallery. Royalty-free under Creative Commons. Download with attribution. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.